Welcome to the Bible and Science. This is Dave Prentice. I'm a retired public high school teacher, certified in all branches of science and math. I've also taught on the Bible and Science for many years, both in the U.S. and on many overseas mission trips. This is episode 32 of the Bible and Science. We've been dealing with DNA, what it does, how it functions, and to me this is such an unmistakable idea pointing toward the conclusion that there's design throughout nature, throughout every living thing. So we're going to continue on with that. I want to recommend to you, there's a good number of videos that I didn't create, so I don't own the copyright. So I'm encouraging you to watch these things. You can go back in the description page for this podcast and you can get those links and you can copy and paste them. They deal with some of the transport mechanisms in the cell. For instance, when something has to be moved away from the center of the cell, it's usually moved by something called a kinesin, which are little walking molecules that carry things away. Then, when something needs to come toward the center of the cell, like raw materials, that's probably going to be brought in by dyneins. And there are also other ones called myosins. If instead of just a very small thing, I have a whole long chain and it needs to get carried away, that's very likely going to be done by a myosin. These are not the only mechanisms, but these are just three of the obvious ones. They need a path to get where they need to go. Well, okay, how are they going to do it? They depend upon something called microtubules, which are little roadways. The kinesin and dynein and myosins, they walk along the microtubules, and the myosins generally pass their cargo hand to hand like a bucket brigade. So please go to those websites and look those up. There's an amazing amount of detail of what's done, and it's probably not 1/1000th of what actually happens in a cell. Nevertheless, it's shown in some really good videos. So if you refuse to believe the idea that there could be design in nature, there are several major ideas. The first one says things can't be designed because that would bring God into science. Well, that's not a scientific argument. That's a philosophical argument. You just refuse to believe there could be a designer, so you won't allow for the possibility of design. In order to disprove design, you would have had to been watching since the beginning of the earth to know that there was never any design involved. So that's the first argument, which is philosophical. Things can't be designed because that would bring God into science. The second one is also philosophical. Things can't be designed because I don't like the way they're put together. For instance, pandas have a bony protrusion on their arm in the place of what would be a thumb, and it's not a thumb. They can't grab anything with it. But what they do is actually use it to strip leaves off of the trees so they can eat them. Well, some people don't like the way that's put together, and they say, because I don't like it, therefore it's not designed. I bought a car from our wife one time, and it had the high beam light indicator on the dashboard so that whenever you turn the high beam on, it was right in your face. I couldn't stand that thing. It was really annoying, so therefore I concluded it was undesigned. That's the same thought process. Just because I don't like the way something is doesn't mean it's not designed. It either means that I don't understand why the designer did it, or I just disagree. But when we start looking at arguments for design, the weakest one is opinion. Somebody says, oh, look how beautiful the sunrise is. There has to be a design. Well, if a person thinks nature is ugly, that's not at all persuasive. But something that's much more persuasive is extreme improbability in a specific pattern. For instance, Dr. Michael Behe, in his book Darwin's Black Box, refers to groundhogs. Well, I live in the South, so I would say armadillos. Suppose there's a busy 1,000-lane superhighway, and we turn loose one of these little creatures, and he's trying to get across. He, because he's got a girlfriend on the other side. So if there's a 1 in 10 chance to make across any one lane, there's no theoretical barrier that says it's impossible for the armadillo or the groundhog to make it all the way across a 1,000-lane superhighway, but we would recognize that's just not going to happen. The extreme improbability will not allow it to happen. But then the best argument for design in nature to me is irreducible complexity. Dr. Michael Behe, and I strongly recommend his book, Darwin's Black Box. Dr. Michael Behe is the one that I first saw coming up with this idea. And if you've got a mechanism that has to have all of its parts in place and properly functioning to do anything, that's irreducibly complex. So Dr. Behe gives an example of a mousetrap. It has to have a base, a hammer, a spring, a latch, and a trigger at the very minimum. If any of those parts are missing, it's not a mousetrap, but it's a pile of junk. Now you can have more parts than that, but you can't have less parts than that. So if there's any part of an irreducibly complex machine missing or not working, the whole thing is a useless waste of materials. So let's consider this when we talk about how to build a living thing. DNA is the building plan for the cell. It's like the blueprints for a house. But if you're building a house, you go have plans drawn up, fine, you got a house. No, the plans are not enough. You need materials like wood, wires, nails, screws, sheetrock, many other supplies. Even that's not enough. Now you need tools to assemble the whole thing, not to mention the fact that you need workers, but that could be humans, it could be machines. But you have to have all of the parts, you have to have the supplies, and you have to have the tools to put it together. DNA furnishes not only the building plans, but the mechanism to bring the materials to where they're needed. It uses microtubules, transport proteins, and so forth to manufacture the tools to assemble the rest of the cell, and DNA assembles itself. It furnishes the instructions for how to assemble itself. So if you look on the bottom, you'll see a model of a typical microtubule. They're similar. These are used as roadways in countless places throughout the cell. They form as needed, and then they're reabsorbed to be used elsewhere as needed. So if you look at the thing on the right, you'll see a cartoonish looking illustration of how a kinesin walks along the microtubule, and it carries things. The little circular thing on the top is like the Jack in the Box logo for one of the major hamburger chains. Well, that's indicating there's something up there being carried along. So the microtubule has to be there, and then the transport mechanism, the kinesin or whatever else, has to be there also. So if you have some parts of the cell that are needed in the outer section, those are carried outward by kinesins. So by all means, go to those links I gave you earlier on YouTube and various places like that, describing how these things operate. They actually walk. Their little feet go flip, 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 flip. So things that are needed in the outer parts of the cell are carried outward generally by kinesins. Things needed in the center part of the cell are generally carried inward by dyneins. All of these have to have microtubules present, and those are present because they're coded for in the DNA, and the places they attach to and the opposite ends, those are also coded in the DNA. Okay, quick recap of how things are moved around. These are just 3 of the major processes. There are countless others, but if The cell has produced something, needs to get it away from the center toward the outer part. It's going to rely on kinesins. If it needs to bring raw materials in toward the center, it's going to rely on dyneins to bring things in. And if they're longer chains, theirs will frequently be transported by something along the lines of myosins, which pass them from hand to hand. But these things all rely upon microtubules, which are programmed into the DNA. The whole transport system is programmed into the DNA. But according to evolution, the earliest cells didn't have anything like DNA. It was just some random collection of chemicals that would gradually evolve into DNA much, much, much later. The error correcting mechanism in DNA is supposed to have evolved due to errors that were not corrected. Likewise, all of the intricate parts of the cell's irreducibly complex mechanisms are supposed to have evolved much later, just one piece at a time. There are many, many, many other mechanisms besides the ones that I just mentioned to you. They're essentially the same no matter how primitive we can just consider the organism or how advanced it is. So unless you're absolutely committed to rejecting the possibility of a designer, if you're a staunch atheist, except for that, wouldn't it seem reasonable to acknowledge that, oh yeah, the similarities, despite all of the difficulties, would point toward design in nature? Pointing toward design points toward a designer, which is going to be God. You know, I'm a Christian. I'm doing this because I believe in God with every fiber of my being, and I'm hoping to give you the same assurance that I have. There are many, many, many other hints of design at the microscopic level. As I've said repeatedly, the first living things are supposed to have had no DNA. It's supposed to have evolved from something along the lines of RNA by accident, and it's experienced so many accidental copying errors that some types of organisms acquired over 100 billion base pairs out of a few base pairs at the very beginning, hundreds of billions. The error correcting mechanisms evolved by accidental errors that were not corrected, and every type of organism known, no matter how primitive or advanced, uses the same genetic code, the same protein manufacturing mechanism of messenger RNA, transfer RNA, ribosomal RNA, ribosomes, endoplasmic reticulum, the transport mechanisms. All of those are the same, but yet amoebas are not the same as humans, which are not the same as whales. All of those things that were producing the same results forgot to evolve. So I just don't think it's reasonable that DNA could have mutated billions of steps from nothing to the present condition, while all of the manufacturing and information storage systems didn't change even the slightest bit. If you're committed to that, okay, well, you have a lot more faith than I do. So just think about how cells reproduce. What would have been necessary? The first cell would have had enough functioning proteins, enzymes, and so forth to be alive in the first place. It would have had to have some sort of a mechanism to hold the information, something along the lines that would evolve into DNA, maybe RNA, and it had to be able to make a fairly accurate copy of all of the parts so it could reproduce. Then once the stored information was ready to reproduce, it would need to be moved around so that it could be used to make copies of all of the needed parts, somewhere along the lines of messenger RNA or something like that. It would have needed a place and a mechanism for the copying to happen. Ribosomes, endoplasmic reticulum, and so forth. Well, why are those there? Because the information to produce them is stored in the DNA. All of the parts had to be present at the same time, or the cell would have become extinct. Life would have gone out. You wouldn't be here. And this is something scientists have discovered Every living thing, no matter how primitive, no matter how advanced, uses the same genetic code. TTG produces tryptophan, GAA or GAG produces glutamic acid, and so forth, as you see on the screen. And the fact that there are several triplets that code for the same amino acid, this allows genes to contain coding for more than one structure because they have to be read in both directions, forward and backwards, to make two copies of the DNA out of a single double strand. I want to give you just a few examples of some irreducibly complex mechanisms in living things. Some of them are really complex. For instance, this is from Dr. Michael Behe's book, Darwin's Black Box, which I highly recommend. Dr. Behe is not a biblical creationist. He's a theistic evolutionist, but he recognizes this couldn't happen without intelligent intervention. Every time you cut yourself, you tend to bleed. Your body is a liquid-filled, pressurized system, kind of like a balloon. But if you have a balloon and you puncture it, it's just going to leak all over the place. Why doesn't your body keep doing that? Why does it stop bleeding? Why does it coagulate? There's a cascade for blood coagulation, and it involves several dozen factors and cofactors and enzymes and coenzymes and so many different parts. If any one of these is not present, you either bleed to death or you die of blood clots. So the proteins that you see on there, this is part of what's going on. You even have Christmas factor. But if any one of these is missing, you can die of blood clots or you can die of something along the lines of hemophilia where you just don't stop bleeding. Okay, so where did this come from? Must have been from lower life forms, right? Well, Dr. Behe points out that all vertebrates have a similar situation to this, and no invertebrates have it. So it just miraculously appeared from somewhere. Well, I would say it's miraculous. It's something that we have to appeal to a supernatural designer for. Here's another little thing that we would say is minor. Antibodies. Antibodies are little flags, and they mark off to your body systems that there's an invader that needs to be destroyed. We can have all sorts of toxins enter into us, viruses and bacteria and all sorts of poisons and so forth. So antibodies can bind to over 10 billion distinct shapes because at the end of the antibodies you have little fingers sticking out like I've tried to show in the illustration there. There are billions of possible combinations of amino acids in heavy chains as well as in light chains. So look at this. This thing can detect over 10 billion different shapes and bind to it and destroy it. Yeah, but you only have 3 billion base pairs in your DNA. How can this happen? Well, I've written computer programs before, and if anybody else has done likewise, you'll recognize, oh yeah, a programmer, it can be a pretty intricate thing. So what happens here is you don't use your whole DNA. You use four actually, small segments of DNA. You have antibody gene clusters, and please go read Dr. Behe's book, Darwin's Black Box. You have antibody gene cluster number 1 has 250 gene segments, number 2 has 10 segments, number 3 has 6 segments, number 4 has 8 segments. So combining in various ways, you can get a light chain and a heavy chain. And the light chains and heavy chains combine in various ways. So one piece of the gene to produce a heavy chain is randomly selected from cluster 1, another one in cluster 2, another one in cluster 3, and one in cluster 4. But the light chain, you use 3 segments instead of 4. And when you combine these, you can produce 10 billion distinct types of antibodies from fewer than 300 gene segments. This is amazing programming. Let's consider some of the safety mechanisms in cells. They often float in a watery environment. And there's a process called osmosis where water will move through a membrane. So since the cells contain a great many chemicals, the surrounding water tries to equalize the chemical concentration through the process of osmosis. This steadily increases the pressure inside the cell, and it would eventually make it burst and die, except that there are safety chambers called vacuoles that periodically release the excess pressure. These are present because DNA has the coding for where to put them, how many to put, what they're to do, and so forth. Well, there would have been no DNA at the very beginning, so the earliest cells would have exploded before they had time to reproduce. There was a tragic situation that happened a few years ago that illustrates this point. A radio station somewhere had a contest called Hold Your Wee for a Wee. People had to drink as much as they could and hold on for the longest possible time before going to the bathroom. Well, there was this one lady who was determined she was going to win this for her children, and so she drank the water and it caused her all sorts of problems and she wound up dying. The radio station had not checked with any medical personnel in advance. They said, oh, it'll be funny, make people hold on and not go to the bathroom. Your body has got a mechanism where the water is going to push its way inside the cell is going to swell, and if you don't release it, it's going to die. And that's what happened to this lady. The vacuoles are a built-in safety mechanism, and she deliberately overrode that. But these things allow excess pressure to be released so that the cell won't be damaged. But in addition to the vacuoles, which are produced by DNA, there's another mechanism called lysosomes. So there would have been no DNA at the beginning. There would have been no way for the cells to release the pressure. They would have exploded. But once a cell dies, it just wastes space and resources. A multicellular organism would quickly become a bag of garbage floating around, except there's tiny suicide sacs called lysosomes. They rupture when they die, and they have enzymes on the inside that haven't been doing anything, but then enzymes do what enzymes do, and they digest the dead cell from the inside out and allow its remnants to be flushed out or recycled. Lysosomes are also present because the DNA has the coding information for how many to put, where to put them, and so forth. There would have been no DNA at the very beginning. If there's no God, if there's no design, life would have quickly drowned in garbage. So could all of these things be the results of mutations, changing whatever genetic information was present on early Earth in the earliest life? And remember, DNA is supposed to have evolved long afterward as a result of mutations in something like RNA, we don't know what. So could these be the result of mutations changing something into DNA for early life? Well, if you read the X-Men comic books and things like that, you'll see that mutations produce super beings. Well, they don't. If they did, then anybody who believes in evolution should be trying to build a home next to a nuclear power plant so they could accumulate mutations and gain superpowers for themselves and their children. No, we instinctively recognize this is not a good plan. Don't try to go eat a bunch of uranium so you can mutate. Mutations don't add any genetic information to the cell. All they do is change the information that was already there. In most cases, this causes damage. If the affected segment of the DNA contains a coding, instructions for a non-essential feature like hair color and so forth, there may not be any obvious harm to the organism. But if the instructions for an essential feature, such as structure of blood cells, organs, heart, lungs, liver, and so forth, if those instructions are damaged, The organism will probably not function normally. It may even die of deadly conditions such as cancer very quickly. Mutations are not good. There are a few mutations that are known to benefit the individual affected, but even though they may help an individual in certain circumstances, they harm the affected species. For instance, here's one of the benefits that's known to have happen in Caenorhabditis elegans, C. elegans. There's a certain mutation that causes it to have more than the normal lifespan, twice the normal lifespan. Yes, but the harm to the species is that the reproduction rate is greatly reduced, so the species suffers. Also, there's a known mutation that affects some birds on the Galapagos Islands. They've inherited a mutation causing them to have small, poorly developed wings. They can't fly, but they're better swimmers. Okay, here's the harm to the species. Put them anywhere else in the world except on the Galapagos, they're going to die. They can't fly. Anywhere else they could not compete with other species. They would probably die off very quickly. Within humans, there's one mutation that's supposed to be beneficial. If a person receives the sickle cell trait from just one of the parents, they'll be immune to malaria. Well, that's great. My mom had malaria when she was a little girl, but she recovered, and it didn't pass on to me. Malaria is serious, but it is curable. It's not added to the gene pool. Sickle cell is not curable so far. It permanently damages the human gene pool. So evolution is supposed to happen to species, not individuals. Even those supposedly beneficial mutations go the wrong way. Why are mutations harmful? Let's take a phrase, in the beginning God created. Could we replace one letter or space at a time to make that say, for God so loved the world? Of course we could. Yes, but what if it had to make sense every step along the way? We couldn't get very far at all. Mutations have to make biological sense. If not, they would produce a corpse rather than an evolving line of organisms. So let's talk about that one mutation known to do some good for human beings occasionally, sickle cell anemia. The hemoglobin molecule in the blood makes it be shaped like a flattened disc. They flow freely through the tiny blood vessels. But if you acquire the sickle cell mutation, it changes the shape of the hemoglobin molecule. The blood cell contains millions of hemoglobin molecules, and it assumes, instead of a flattened shape, a sickle shape. This can lead it to catch and block off the small blood vessels. Sickle cell prevents oxygen from reaching the cells. An individual receiving the mutation from both parents will likely die a slow, painful death. The benefit, yes, if the individual receives a sickle cell trait from only one parent, he or she will be immune to malaria. The harm to the species is that sickle cell is permanently added to the gene pool. It doesn't help the species evolve, it damages it. So this is what makes sickle cell happen. You have several billion base pairs on your DNA. One single DNA copying mistake causes sickle cell anemia. Normal hemoglobin is shown up there: leucine, threonine, proline, glutamic acid, lysine, serine, and the DNA sequence is shown below. Sickle cell has one amino acid different. Instead of the first glutamic acid shown in red, it substitutes a valine shown also in red, and instead of GAG, it does the sequence GTG. The substitution of that one thymine causes sickle cell anemia. This is because of one place out of 3 billion in the DNA. A person could go for years and barely scratch the surface of what's going on in living things. We've talked a few things about what happens in the cell but we're going to gradually move up more toward the macroscopic level where you can see visible things throughout nature. So I hope that you consider this to be reasonable, that yes, there could be a God, there could be a designer. See you later.